Cheers.
I know.
The second half of the year. She's like, what's the occasion? I was like, at the game today. She's like, oh, who's it against? I was like, Breck. She's like, oh, that'll be good.
Well, she really roasted you.
Because I think she was being sarcastic. She said that. I was like,
What are you doing after school? Are these active notes?
Yeah. I thought
Yeah.
I don't think yesterday they were.
Oh, lucky you.
the topic.
Oh, that's because I wasn't sure if I was going to go back and re-lecture, so then I just gave you all the notes. So yesterday's were not active, but today's are, which I just posted this morning. Okay, so we will not be able to extract DNA today because yesterday I was like, oh yeah, I need to print off the directions for everybody. And I must have went back to the printer. I went to the printer after every class period to go see if they actually printed, and they never printed. And I didn't get it figured out until after school yesterday. So after Mrs. Oaks had left, basically. So we'll extract DNA tomorrow. It only takes 5 minutes. You just spit into a test tube. You add some meat tenderizer that breaks down the cell membrane so your DNA gets free and liberated. And then you add alcohol, and then it causes your DNA to cloud up and clump together because there's an electrostatic force between DNA and alcohol. They're kind of like opposite charges. And then instead of taking a wooden skewer, because I don't have any, you're going to use a noodle and just kind of wrap it around, or maybe break the noodle in half in Chinese. What do you call it? Chopstick it out and look at it. And yeah, so it only takes about 5 minutes. So without further ado, then I guess I'll just lecture on Chapter 14, which is the genetic material. It's all about DNA. We'll talk about the discovery of DNA, what it's made up of. We'll talk about how DNA replicates, the differences between eukaryotic and prokaryotic, and kind of finish the section talking about telomerase and how it ties into aging and cancer with it. So with previous work by Gregor Mendel and Thomas H. Hunt, we know that there were chromosomes, but we didn't exactly know which part of the chromosome actually contained the genetic information that carried the traits and the alleles. We knew that chromosomes are made of DNA and a protein, but we didn't know if it was the DNA or the protein that actually carried the genes. So scientists went out to try to figure out, is it the genetic material, which we didn't know at the time it was, or the protein that has the genes. And so Avery Griffith was studying bacteria cells, and he noticed that some bacteria cells can be transformed. He was studying a specific strand, Streptococcus pneumoniae, which causes pneumonia in mice. And there's 2 forms of this, a normal virulent form and a mutant form. And so with this form, it actually caused the disease, pneumonia. He called it the S form for smooth. And then there was this mutant form, which means that it was not dangerous, okay, it did not cause the disease. And it was called an R form because the surface of it was rough. And so he was studying these 2, injecting these forms into the mice, and he kind of noticed some discoveries or observations. So with, I'll just kind of break down his experiment here. So up top, we have the living encapsulated bacteria. Okay, the bacteria is inside this capsule, and he injected it into the mouse. The mouse died. Okay, then if he took the R, so this is the S form. So then if he took the R form, which is like the rough, and it was alive, however, it did not cause the disease. Notice it doesn't have a capsule. Injected it into the mouse remained healthy. Nothing affected the mouse. It was normal. Now, if he took the S form from 1 and he heat killed it, so they changed the color. So this is kind of like this dark peach color, and this is less lighter peach color. So he heat killed the bacteria, injected it into the mouse remained healthy. So he said, okay, the S form is dead here. What happens if I take the R form that's alive, that was healthy, and mixed it with the S form, which is dead, and the mouse was still healthy? What's going to happen there? So he did that. And what he discovered was that the mouse died. And he's like, wait a minute, how's that possible? Because it lived here and it lived here. Well, it should live here. Well, then he realized that the bacteria transformed. It went from dead to alive. The dead S form took on parts of the R form and kind of came back to life.
So even if you kill the bacteria, it can still come back.
Yeah, by taking in kind of like dead parts or bits and pieces of the other form. So he took samples and he grew it in culture, and he realized the colonies were just right back to the virulent form that actually caused the disease. So he's like, well, how did the dead S transform back to alive? And it had everything to do with DNA. Wait, I'm just waiting for Logan to come in. See if we'll dress
Yeah, we
up.
should have known that if he's usually 10 minutes late, then he'll
He
today
brought him
he's going to be
past
about
yesterday.
20 minutes.
I was
Okay.
like, yep, it's the first part of the month.
So Griffith called this process transformation, where a virulent form kind of like took in parts of another and kind of went back to its original state. So here we have this bacteria chromosome, and bacteria can take in foreign DNA in its environment and incorporate it into it. So we call this stable transformation if that occurs, or if it doesn't incorporate it into the genome, now you just have bits and pieces floating around the cytoplasm. So Avery, McLeod, and McCartney actually identified the transforming principle back in 1944. So, close to 20 years later, they, picked up where Avery left off here, prepared a mixture of dead S and live R streptococcus, removed the protein, achieving 99.98% purity of genetic material. A little bit of protein, but they got really close to extracting all that protein, and the transforming principle was not reduced, which means that the S form came back alive. The transformation took place. And so they said, hey, we got rid of the protein, all that's left is the genetic material, so that's the transforming principle. But the problem was, no one believed them. They had all this evidence. So the properties of these substances resembled those of DNA in several ways. The composition was very closely to DNA. They had the same density as DNA. The lipids and proteins did not reduce the transforming activity, which means that the R or the S form came back alive. Protein digesting enzymes did not affect the transformation. So after they cultured it, they added protein digesting enzymes to see if it would actually break down. It didn't break down because it's not protein. They added DNA digesting enzymes. Then it destroyed the transformation process. So they're like, hey, it's got to be DNA and not protein. So here's their evidence. This is actual data taken from their experiment where they have multiple samples, okay, and they're comparing it to the known value of DNA, and you can see that they're relatively close to DNA, the ratio of nitrogen to phosphorus. Yeah, all of their samples really, really close to DNA, so why can't it be DNA? But again, the scientific community was not ready to accept the fact that it was DNA. So it wasn't until Hershey and Chase demonstrated how genetic material is DNA working with bacterial phages. So Avery's results, they were not accepted. They still believed proteins carried that hereditary information. So Hershey and Chase looked at viruses that infected bacteria. And so viruses that infect bacteria, we call them bacterial phages or phages. So the phage that they used was kind of scary looking, okay? It's got this capsule, okay? And then I like to call this like the syringe part, where it's got a needle at the end and it's got these spidey legs. And so what they do is they attach to the surface of the bacteria cell, and then they pierce through the bacteria cell walls, because it's the cell wall is made of peptoglycan, so it can be pretty thick. So it pierces through that cell wall, and then it takes its DNA and inserts it into the bacteria cell, so that way that DNA, viral DNA, gets incorporated into the bacteria's genome. This is how viruses work. Viruses, they're not considered living things. They have to hijack another cell's machinery to make more viruses. can't do it on themselves. Virus is basically just a protein coat in DNA. It has no ribosomes, doesn't have any other organelles. It's got to hijack another cell to do all its dirty work, basically. So in this actual picture, you can see bacteriophages are attached to the surface of the bacteria cell. And these are small. I mean, bacteria cells are really, really small. Take a look at these phages, even smaller. And you can kind of see these green lines here of the phage DNA being inserted into bacteria cell. So it's kind of cool. So here was their experimental setup. They took these phages and they labeled them using radioactive isotopes. On one part of the phage, they coated it with sulfur, the protein coat, and then in a different experiment, they labeled the DNA. So they took these phages that had the sulfur coat, and they mixed in with some bacteria phages to see how they'd interact. And you can see that they did their thing. They infected the bacteria cell. And then at the very end, they looked at the culture, and they realized that all the fluorescent dots were outside the cell. So they said, hey, protein is not inside. Protein is not the transforming principle. In this experiment, they had labeled the DNA. And so the bacteriophages affected just like they did in the other experiment. And then they looked at the bacteriophages at the end, and they realized that all the fluorescent dyes lit up inside the bacteria cell. So now they actually had picture evidence stating, hey, DNA is the transforming principle that is being transferred, that carries the genes. It's not protein. And then after all of that, then they finally accepted the fact that DNA is the transforming principle. So let's talk a little bit about the DNA structure, which will be slightly repeat here. So DNA has three main components. You have a phosphate group, a 5-carbon sugar, and then a nitrogenous base. The sugar in DNA is called deoxyribose, and in RNA they call it ribose. The nitrogenous base is what makes the nucleotide different from each other. So the phosphate group and the sugar are usually the same, but it's the N base that makes the 4 different nucleotides. So we talked about purines, adenine and guanine, angels and gods are pure. That's our little saying. Then we have cytosine and thymine in DNA, C and T. They are the pyrimidines. All right, so this next little section is going to talk about, we know DNA is the transforming principle, but what's the structure? This is what it is, but we didn't really know that at the time. So again, another diagram is showing the difference between purines and pyrimidines. Purines, double ring, pyrimidines, single ring. How nucleotides bond, and I feel like we've discussed this too. It's between the 3-carbon and the 5-carbon of the 2 nucleotides. Okay, so here's the 3-carbon, here's the 5-carbon. So it's the 5-phosphate linked up to that 3-hydroxyl, and we remember our dehydration synthesis means that we take water out to form the bonds. The bond between nucleotides here, like between one nitrogenous base and the next in the rung of ladder, is a phosphodiester bond because it involves that phosphate, and there's 2 of them. Linear strands of DNA and RNA have a free 5 end and a free 3 end. DNA always runs in a 5 to 3 direction. So if I were to add another nitrogen base to it, it's going to go onto that 3 end. Okay? So it's built from 5 end to 3 end. And the reason why I'm emphasizing this is when DNA replicates, it always is made in a 5' to 3' direction. So here's a diagram showing the formation of the phosphodiester bond. Here's my 3-carbon. There's that hydroxyl group. There's the phosphate attached to the 5' carbon. And we're going to release water, okay? And now we have a phosphodiester bond. Chargaff, Franklin, and Wilkins kind of obtained some structural evidence of DNA in its early stages, and then we'll talk about how Watson and Crick stole, borrowed, whatever word you want to choose, to actually figure out how it all works. But Chargaff was looking at nucleotide composition of DNA, And he noticed that the number of adenines always equaled that of thymine, and the number of guanine always equaled that of cytosine. So they're always in these equal proportion of purines, A and G, as well as the pyrimidines, C and T. So we call it Chargaff's rules, where A equals T and C equals G. Franklin was working with X-ray diffraction, and she's the first person that kind of took a picture of DNA using X-ray diffraction. She didn't really know what she was looking at, and she just knew that this picture was really important, so she started to share it with like one scientist, and this other scientist knew Watson and Crick, and Watson and Crick obtained this photograph and used it without her permission, and so they're the ones credited with the structure of DNA, even though she was probably the one that took the picture. Without it, no one would have known the structure of DNA. But it's bombarded with X-rays, rays are bent, they're diffracted. This is the actual photographic film, and it suggests a helix shape, kind of this twisted corkscrew. So you're kind of looking at DNA from the top, and so you can kind of see the rungs of the ladder and all the little the rungs, the staircase, if you will. So Watson and Crick took all that evidence that was available, and then they didn't do any experiments. They just like took everyone's evidence and then, like a jigsaw puzzle, put it all together. And so they built the DNA structure out of wood, and you can kind of see here ring stands and ring clamps to show there's a 3D model of DNA. Okay, so DNA has obviously a sugar phosphate backbone, and then the nitrogenous bases that go across and link to each other. And remember Chargaff's rules, A equals T. That's because A always pairs with T. And so that's why you always got equal amounts of A and T and C and G. Another thing is, you notice we have like 3 rings across. 1, 2, 3. 1, 2, 3. 1, 2, 3. And so that's why we always have an adenine or a purine bonded with a pyrimidine. So this is a purine bonded with a pyrimidine. Here's a purine bonded with a pyrimidine. And so it's got to fit. If A paired up with T, well then we'd only have 2 rings, and if A and G it'd be 4. So your DNA strand would be like this instead of a nice ladder, if you will, same width apart. These dashed lines represent hydrogen bonds. So there are 2 hydrogen bonds that hold the A and the T, and there are 3 that hold the C and the G themselves. So we have complementary base pairing, where A always pairs with T and C always pairs with G. Here are the number of hydrogen bonds, 2 hydrogen bonds between A and T, 3 between G and C. And again, purine always bonds with pyrimidine to keep the diameter the same and to obey Chargaff's rules of proportion. Another thing about DNA is it is anti-parallel, which means if we have 5' to 3' in one way, well, that means we're going to have 5' to 3' in the opposite direction. So both strands run anti-parallel to each other in opposite directions, where one strand is the 5' to the 3, and then if you're going the other way, 5' to 3. Any questions on the anti-parallel? Okay. All right, DNA does replicate. It has to replicate in order to divide, and so basically it has 3 steps. The first step is initiation, the start of it. Where does it start to replicate? So we call this the origin, and the origin of replication is called the OR. So if you ever see OR capitalized on a diagram showing DNA replication, that just means the origin of replication. We have proteins that come in, they bind to it, the origin, and then it starts to kind of unzip the helix, exposing those single strand templates. So it unzips, and then we have single strand templates that come in and match up on both ends of the zipper, building new complementary strands. So we have initiation. The second step is elongation. The protein or enzyme responsible for that is called DNA polymerase. So DNA polymerase binds to the open strands, and then it takes those nucleotides and match pair it up. DNA polymerase does have some common features. It always adds bases to the 3' end. So that means DNA is made from 5' to 3'. It does require a primer to bind onto synthesis, and we'll talk more about that primer, I think, on the next slide. It cannot begin without a strand of RNA or DNA base paired to the template. It needs to start with a primer. So here is my OR, my point of origin, and it starts to unzip. Okay. Then we have these single-strand binding proteins that latch on to keep it from zipping back up. And then we will have our DNA polymerase latch on and start reading the strands, and then these free nucleotides will come in. I should also mention some other proteins in this diagram here. So here we have DNA helicase that actually does the unwinding of DNA. And to help you remember what DNA helicase does, there's this dirty, dirty joke out there that, you know, it's a horrible pickup line. So like a guy says to a girl, "Oh, I wish I was DNA helicase so I could unzip your jeans." And so it's a dirty joke out there. But help you remember, DNA helicase unzips DNA. We also have some proteins that help with the twisting, if you will, of DNA, because as you like unzip, I don't know if you've ever like taken a braid out, but if you were to like pull on this, well, then you're going to cause a lot of torsion up here. So we have like DNA gyrase that kind of helps reduce the torsion, if you will, of the replication fork, so it unwinds properly and doesn't get knotted up. Okay, the third step is termination. And so in prokaryotes, remember their DNA is circular. So then if it starts here, it goes bidirectionality, and then they'll meet somewhere over here and then be done. Eukaryotes, they are linear. And so they may start at multiple points and like work towards each other, but it's halted at the ends of the chromosomes, and we call the ends of the chromosomes telomeres, which basically just means repeated regions of bases. So in prokaryote, it's a lot easier, less complex. Here's my point of origin. It goes around bidirectionality. They'll meet at the opposite ends, and now you have two identical circular DNA strands. DNA unwinds by the enzyme helicase. It does require energy from ADP to unwind that DNA template. When DNA unzips, we have these things called SSBs, or single strand binding proteins, that bind to it to keep DNA from zipping back up. So then here's my DNA polymerase, and it starts to read the base pair, and it starts to match it up appropriately. And then once it, reaches this area, the SSB will fall off, and it just keeps reading the code. So these SSBs, they just prevent the DNA from zipping back up. I also mentioned that the unwinding can cause torsional strain. So we have gyrase, but we also have something called topoisomerases, which literally cut the DNA, untwist it, reattach. Cut, untwist, reattach, to reduce torsional strain as it unzips. So lots of nitty-gritty players in the process of DNA replication. Replication is semi-continuous because DNA is anti-parallel. DNA, what direction is DNA made? 5 to 3. So when the DNA polymerase, I'm just going to abbreviate DNA poly, when it reads that strand, one strand is continuous because it's in the right direction. But the other strand, we have DNA polymerase.
It's like choppy, right? Or something.
It's very choppy. So it reads until it reaches, until it doesn't need to read anymore. So if it started here and it started to code here, well, now DNA has unzipped more. So then it will fall off and reattach in an open spot and read it again until it reaches a section that it has already replicated. So they call this continuous, but this, it stops, has to fall off, reads it again, fills in, has more DNA, is unzipped. So we call these Okazaki fragments.
Where did we learn about that? Did you teach us that?
We
I didn't have
about
you-
it last
did-
year in Rhodes.
Yes, you guys taught it from Roards. I didn't have you for biology, did I?
But
So-
is it reading it from 5 to 3? So
It's still, yep, so it's reading in 5 to 3, but it's being like unzipped in like this direction.
well, wouldn't that essentially do the same thing if it's 5 to 3?
It is the same thing, it's just that it's doing it in parts instead of continuous like the top strand. Yep. So we call the strand that's continuous the lead-in strand, and the strand where it's made up of fragments, the lag-in strand, because once it's, completed a section, then it falls off and then has to like reattach and start all over again. So it's just bits and pieces work together. So this diagram attempts to show the Okazaki fragments with the continuous strand. So here's our lead-in strand. We're building it in a 5' to 3' direction. It's continuous. Yay! These are supposed to represent the SSBs, the single strand binding proteins that prevent it from zipping back up. Here we have helicase, and they picture it as a little wedge to unzip. Here we have an RNA primase laying down the primer because DNA polymerase can't bind unless there's a primer laid down, so green stands for primer. So this DNA polymerase binds to the primer, and it started to make the strands, and then once it reaches the primer of another segment that it had already completed beforehand, it will fall off, and then it'll latch onto another primer and fill in the missing blanks until it reaches another primer and fall off. So the primers act as starting points as well as ending points.
Primers are only on the lagging strands then.
There will be one primer strand for the start because you need a primer to start, but this will have many primer strands, which is okay. Like it still codes for the genes. It's just
And what's the length? How long would it take for them to pull the whole or unzip and
your cells can do this in like 20 minutes. I mean, it doesn't take long because it has multiple starting spots
But
on
it
a
is like
chromosome.
20 minutes though. It's not like a nanosecond.
Oh, I don't know. I don't know. Because in the process of mitosis, cells can divide in like less than 20 minutes. So like, yeah. Good question.
It's weird,
Sorry.
because everything else is so fast, so I would have guessed like this happens like whole mitosis.
Oh, I'm
process.
sure.
I would have guessed that happens in like 10 seconds.
I'm sure you could Google like how many nucleotides does DNA polymerase like bind, and I'm sure they'll give you a pretty estimate number, like a pretty good estimate number. Because you can do this in cultures, like you can put in 100 nucleotides, stick in one DNA polymerase, and time it before it uses up all those nucleotides. So I'm sure they got a number out there.
100 to 200 nucleotides on the lagging strand. Primers are about 10 nucleotides long.
Okay.
So 100 to 200, I'd say, on the lagging.
Okay, so eukaryotic replication is a little bit more complex because we're eukaryotes, so we have more stuff to us. But our chromosomes are linear, and so when your chromosomes replicate, they're doing this. We have multiple origins, and you can kind of see here that, yeah, we have, helicase work in here, helicase work in here. I think this looks like a little one here. Yep, cartoon diagram shows me up there. So they work towards each other to get the work done faster. Linear chromosomes have a different termination. So in like prokaryotes, they'll meet at opposite ends because their DNA is circular. But because our chromosomes are linear, at the ends of the chromosomes we have telomeres, which have specific DNA sequences. Yep, so telomeres protect the ends of the chromosomes and they help maintain the correct, all the genes. Because on that Lagan strand, the Okazaki fragments, eventually you're like every time your chromosomes replicate, you're going to lose a little bit of nucleotides. So these telomeres that hang out on the end, they're just repeated nucleotide sequence that kind of don't code for anything. So it's okay if you lose a couple nucleotides every time it divides. So I'm going to come back to telomeres with aging at the end, and I'll actually show you a chromosome in the embryonic stage and then as an adult stage. And you'll see that the telomeres shorten as you get older. So they believe telomeres are tied to aging.
So what if we eliminated the loss of?
If you could eliminate the loss of telomeres, yeah. So possibly reverse the effects of aging. The guy that went to space for a year, it wasn't quite a year, but it was close to a year, he had a identical twin brother, and so they compared the telomeres between him and his brother, and his were shorter. So they think something about space and
Makes you age less.
No, age faster.
age faster. So replicating the ends, remember, we have that bidirectionality where one strand is continuous, the other strand is Lagan, and the primers create a problem for that replication because it's going to leave a gap, and it's going to be primed and finished, and it's just going to you're just going to lose a little bit of nucleotides over time. So we call our telomeres the kind of useless strands, and I shouldn't say useless, they're important, but they do shorten over time with each round of replication. So here's a diagram showing the ends of the chromosomes, what we call the telomeres, okay? And we have an enzyme called telomerase that, basically kind of makes the ends work here. So it says the repeated nature of the short repeated sequences of telomeres is the result of the enzyme called telomerase that you can kind of see in that top right-hand corner. Telomerase uses internal RNA and not the DNA itself as a template. It allows short stretches of DNA to be synthesized without primers. So when it gets to the end, telomerase kind of takes over. However, we do lose a little bit of telomeres with each round of replication. So here it is with aging during embryonic and childhood development. Telomerase activity is high, which means that it adds on, like it lengthens it. But then as you get older, telomerase activity decreases. And so then your telomeres get shorter. So then what they're trying to do is they're trying to, mimic this in lab. If we just keep adding telomerase to it, what's going to happen? So the telomerase activity in cells that's kept low by preventing the expression of those genes. Lack of its activity and shortening of telomerase is related to aging. And so, again, looking at telomerase and that aging process. Now, telomeres are also, they believe, connected to cancer. So here's a picture showing embryonic stem cells, and you can kind of see that the telomeres are a lot longer. And then as you reach adult and we look at our stem cells, they're a lot shorter. So what's going on here? Our telomerase enzyme is inactive or absent. It's stopped adding on the ends.
So when it gets to none left, is that like a sign of like when you die?
Possibly, like when things start to go wrong,
That'd be
you
when
get
your-
cancer,
that'd be when your
tumors,
cells
syndromes.
stop, replicating then, so
I don't know if they would stop replicating, it's just that they would replicate, but you'd be missing very important information, which would cause defects, which would cause health problems.
Maybe that causes what's the thing when you start to lose your mind, kind of like
Dementia.
maybe.
Yeah,
Alzheimer's. Yeah. That's my theory, Mr. Erickson. This causes Alzheimer's.
Okay.
It's a breakthrough.
Alzheimer's has something to do with plaques in the brain, so possibly
We're
because.
finding out information on what causes it.
I just solved it.
So they're trying to look at the telomerase, which is the enzyme that adds on telomeres at the end of your chromosomes in cancer. So they've introduced telomerase to cells in culture, and they've noticed that you can increase the lifespan of those cells because it doesn't lose information, so that they don't, get defected towards the end. These cells did not become cancerous. Cancer cells do show activation as telomerase, So for some reason, cancer cells have high telomerase activity, and that's why they just always like continually divide and divide and divide and divide and divide and never slow down.
So can we do that to normal cells? Like,
Well, that's what they're trying to do. That's what they're trying to do.
If they got smaller, could we just get like a micro, like a macro, tiny 3D printer and just print telomerase and just attach it?
or can't they just like slow down the activity of the cancer cells?
Yeah, definitely. I think it's got many applications, but again, it's hard to do this in humans because
It's got to go through all that.
it's got to go through all those trials and clinicals before you can do human application.
That's Erickson. So right now the problem is that the cells have their normal rate and the cancer's too fast. What if, you know how they do this and they like sped up the cells? What if then the problem just became some going too slow? You know? Kind of like, you get what I'm saying?
Yeah, because then you could prolong it
It's
and
like the opposite of.
that'd be cool. All right, now DNA, we've talked about how DNA can repair itself. So with DNA replication, there are some checkpoints inside the process of the cell cycle where it looks for mistakes and tries to fix it. We talked about the P53 gene, and it goes back and tries to reverse the errors. So we do have systems in place to repair some damage. There are many, many repair systems that have been found inside of us. All cells show many ways of repairing DNA and reversing those errors during replication. So I'm just going to talk about a few of those repairs. So one repair is called a photo repair, or UV light can cause thymine dimers. So here on your DNA, you can have two thymines right next to each other. And when UV light hits the strands, sometimes the T's actually bind it to each other instead of from across the nucleotides that they're supposed to, in this case, two adenines. And so here we have this dimer where it's bonded to itself, and these two nucleotides, they're free and they're not, being paired up to their correct nucleotides. So then when DNA polymerase comes along and it reaches here, they're like, what the hell, I don't know what to do. And DNA may not get replicated correctly. It might just code for one A instead of two A's because of it. So one way that it can correct the dimer is using an enzyme, photolase. So you can kind of see that we name enzymes after what they do. Lipase. So ASE stands for an enzyme. So here's what DNA should look like. Here's my thymine dimer. They denoted it as two little reds and they're stuck to each other. So this enzyme comes in and reverses the effect, cuts the bond between the two, and forces it to bind to the two A's across the way.
So the purple's like the sunscreen blocking the UV.
Yeah. Well, it's blocking the UV, but it's also repairing it. It's cutting that bond between the two thymers. So I'll go back. Photo, we call this photo repair. UV causes the dimer bond, and then it actually uses light to fix the problem with this enzyme. You can also do excision. This is usually a non-specific repair, so that was a specific. Two thymines bonded to each other. With excision, they can kind of cut out or replace the damaged part. So basically 3 steps. It recognizes there's some damage, it removes the damaged region, and then it remakes the damaged region and fills in the blanks as a template. So here is a diagram that I think does a way better job than my words. Here's the damaged part. Well, maybe we're not going to use photolase to repair it. Let's just cut it, get rid of it, have DNA polymerase come back in, fill in the blanks correctly, and repair the damage. So this is non-specific. All right, so that's chapter 14 in a nutshell. Tomorrow we'll extract your DNA, like I said, late. It will only take like 5 minutes, so just be ready to spit. And then we can review. I don't know if you've noticed, but on Google Classroom, I made a flowchart of chapter 14. So I'll just pull that up and kind of discuss it with you. Or maybe I didn't. Never mind. I'm thinking of something else. I will upload it right now. Never mind. So I'm going to post this onto Google Classroom. It's a flowchart of Chapter 14 in a nutshell, or a concept map, and it kind of tells you what I just discussed today. So I started with Frederick Griffith and the mice and his experiment, and how he was not accepted by the scientific community until Hershey and Chase, and then they believed him. And then it kind of cuts off here. So we'll say what we know. So we know, yeah, very artsy fartsy. So I talk about what Franklin contributed, what Watson and Crick contributed, the process of replication, how DNA replicates, the little enzymes involved, how DNA gets repaired. So it's just like a summary. I'll do this also for the next unit, because the next unit can get pretty difficult, nitty-gritty details. So it just kind of helps you organize your information. But